showed that from 3,300 years up to 2,400 years, this area was mostly dominated by herbs of Cyperaceae, Poaceae, and Brancaceae, showing that during this time there was open landscape in the surrounding of in this coring area. From 2,400 up to 900 calibrated years, here there is a decrease in Cyperaceae, but with the decrease in Cyperaceae there is an increase in Poaceae. From 2,400 up to 1,500 years, Cyperaceae decreased and Poaceae increased. This means that the wetter climate was replaced by a warmer. The colder climate showed a shift from cold to the warm, with the spread of Poaceae in the area. And also taxus and cedrus along with abies appear prominently in the area along with this, and also among the trees and shrubs the quercus, jubilans, and bitula appear. But if we look from 900 up to 300 years, the area became very rich in the vegetation. The herbs spread in the area along with the trees and shrubs. Along with on the surrounding mountains, the conifers, Pinus, Picea, abies, cedrus, and taxus, they spread in the area, and which shows that this is the time when there was sufficient rainfall in the area, which resulted in such rich vegetation during this time. During the last 300 years, we see that conifers have spread enormously in the area. That is, we looking at the peaks of Pinus, Picea, abies, cedrus, and taxus, and the trees and shrubs are consistently present. And during this time, barbarus appeared in the vegetation. Among the trees and shrubs, the taxa shows the same level of their spread in the area. And here, with the decrease in Poaceae, Cyperaceae has increased. So if we combine this appearance or decrease of Poaceae, which shows that the grasslands are shrinking and the conifers are spreading during the last 300 years in the surrounding of Kabul Swat. What we conclude from this work, that polar record shows the vegetation and climate history of the last 3,300 years of the late Holocene. From 3,300 to 2,400 years BP, the dominant herbaceous vegetation was replaced by taxus, Pinus, jubilans, Poaceae, and Cyperaceae. The decrease in Poaceae from 2,400 to 500 calibrated years, and again an increase from 1,500 to 1,200 calibrated years, shows that Kabul Swat went through respective wet, cooler, and dry, warmer periods. The abundance of Poaceae, Cyperaceae, Brassicaceae, Mardancaceae, and Asteraceae from 900 to 300 calibrated years before present shows widespread deforestation and agricultural activities during this time. Conifers like Pinus, taxus, Picea, abies, and cedrus have expanded during the last 300 calibrated years. These conifers mostly occur in the mixed coniferous forests at higher elevation in the Alpine area today. Here is some future outlook for extending my PhD work. When I go back, I want to establish a paleoecology lab at my parent university, where I serve as lecturer in botany department. For my first sub-project, the data set is limited, and my next target is to collect at least 5 to 600 species of Poaceae family to know more about the pollen grain size variation friends in this family. And I'm also more interested in looking into the pollen grain size variation in C3 and C4 related or sister taxa. So I'm already in contact with a Brazilian group, and we will be sharing data from Pakistan and representing South Asian species, and this person from Brazil to look into the South American data sets. For my second sub-project, next I want to increase the number of plots and their sizes. And also I'm interested to establish vegetation and pollen relationship at genus and species level in my research area. And for this project, I have already, I'm in contact with the Bilsani Institute in India and one of my friends in Afghanistan, and we want this study to extend it to Afghanistan and India and my region. For my third sub-project, I am interested to take more core from the lower grasslands and upper Maranjaba mountain, because this is the first study in the area. So I'm interested to take more cores, and I also am interested to establish collaboration links with international paleoecology labs, because the techniques which I learned here in the paleoecology, I'm hopeful that I will be able to start with paleoecology work in my university. But the limiting factor in my mind is the financial support for my radiocarbon dating, and for this I think that I want to establish links with the international paleoecology labs. Because the human resource is sufficient there, we will work there, but for radiocarbon dating we will be needing financial assistance, and that's what I'm planning for. So I'm thankful to the financial support of my university under the Faculty Development Scholarship, my department, the faculty, and Göttingen University. I am thankful to my supervisor, Professor Herman Wieling, and my co-supervisor, Dr. Lisa Schuller, my research group, the department members, and the person who helped me right from day one up to the end, Dr. Georg, and Uta helped with in the lab, and Dr. Wahid Murad, ex-student of Professor Herman Wieling. He spent night with me in the field. I'm very thankful to him. The graduate figure in charge of the botanical garden who helped me a lot, and Dr. Digo from systematics department, and Professor Wali from Brazil who gave me valuable suggestion. Dr. Naveed from vegetation department, my master supervisor, Dr. Jahandar Shah, and my friend Dr. Zahid, assistant professor of conservation biology at Swat University. I'm thankful to, I cannot name all the persons, but I'm thankful to all. That's it.
Thank you very much for your clear and nice presentation. We have time for questions. So who wants to start?
I would also like to thank you. I mean, I have a series of questions, but I would start with the first simple one. You wanted to distinguish C3 and C4 plants, and I wonder, do you need the pollen afterwards? And if not, why not simply mill them and look into, let me say, the relation of carbon isotopes?
Can you please repeat the question?
Yeah, I'm interested in many things, but also in sub-project number 1, and it was about separating pollen from C3 and C4 plants. And I wondered whether, at least if the financial substance and analytical capacity is there, if it would not be a possibility to just analyze the carbon isotopic composition of these things as they follow different photosynthetic pathways. They will have a different signal in the 13C to 12C signal. Is that a possibility?
much idea about the isotopic studies, as we know we are pollen people, we work with pollen. And here the idea is to look into pollen grain size variation in Poaceae species growing in different climates in different regions. And our question, basically, we were interested to know whether is it possible to classify grasslands growing in different climates based on the pollen grain size. That was the question, that was the idea. And in our results, we established that yes, though the scores were not clearly separable, there was overlap, but there were trends. And based on those trends, we can look at, okay, what are the increasing and decreasing trends of pollen grain size of Poaceae species collected from a certain grassland in a certain climate. And the results we get, okay, what you mentioned, the evidences from isotopic studies and also from the phytolith, because there are groups who work with phytolith assemblages. So we can then compare our results with their results and try to reach at more clear conclusions as whether is it possible to classify grasslands, because this project basically is a pilot project. It is only a 160 species, and I mentioned that it represents only 1.45% of the family. So, but the next target is that I'm interested in increasing this number. I had no idea by that time, so I worked, from my transit, I just collected, when I started my PhD, I started collecting species. Here, when I brought them to Göttingen, Professor Billing told me the data set is not sufficient. But by that time, I didn't have even idea to look at the look at this trend in a certain related taxa, in a certain like taxonomic focus, a certain taxonomic ring. But this now is my idea that next this is my interest. That not only I want to increase the number of Poaceae species and look at this trend, that whether how comes the result, and also I'm interested to look into focus a certain taxonomic range and look at the related C3 and C4 taxa, which sort of trend they show. But this is my interest, and in the future I want to work on it. Okay.
My question also refers to the first sub-project, and as you say, you are interested chiefly in the research question of how and to which extent in a certain region differences can be found between the proportions of, let's say, C3 and C4 grasses. Then why do you use the whole data set, I mean a worldwide data set, for such a regionally regional interest? Why didn't you use your assortment of grasses in subsets of regionally occurring grasses? It would be more logical if you are interested in knowing the C3, C4 ratio, say in Pakistan or in Africa or in South America, then you should have used each of these partial data sets.
Okay, so as I told that the data set from my research area, from this 200-kilometer gradient, where I started from the plains and went up to the upper mountains, this data set was small. So one was this a limiting factor to that later on when we will have results, the data set was extremely small. Then the next question which could arise was, even if I arise at certain conclusion that okay, in this climate this is the increasing or decreasing trend from grasslands in the species collected from grasslands and species collected from the mountains. But even then, the next question was, can this effect be due to the climate in this particular area? Is this trend, can this trend be seen in other areas also or not? Because the area may be due to certain climatic effect or drastic climatic effects. So therefore we went after extending our spatial scale so as to know, because we were more interested in the family, and the family exists everywhere. So the results should justify not only my research area, but it should also justify other areas around the world. So that was the idea, and like the data set was small, and the next thing was to see this effect in Poaceae species on larger spatial scale, because to include grasses growing in different climatic regions and then see. So that was the idea.
Yeah, I think we have also addressing these 3 4 points. In your presentation, I missed the underlying hypothesis why the pollen grain size of C4 grasses shall be, or is hypothesized, shall be bigger than the C3 grain pollen grain size. What is the underlying hypothesis for that? Why do you think that this is the idea, and why? What is the difference between the C4 and the C3 plants, which also affect pollen grain size? I missed this hypothesis, or the explanation for this hypothesis.
Well, so this it is already established. It's not my claim about C3 and C4 species. C3 and C4 species in the rest of the plants as well as in Poaceae family is already scientifically established fact that they exist, and that is not my area. So initially when I started the work, I started counting the chromosomes, and I came here to systematics department and I started work on that. But then later on I realized that I am entering into a different area which is not my expertise at all. So, and that was leading me somewhere that probably I would have spent the rest of my 3 or 4 years of my PhD counting the chromosome and determining the diploidy level of a certain taxa which I had collected from the area. So I relied on already published data regarding diploidy, and I had no other option than to go for that.
Okay, may I add to that point? If I got you right, so the pollen grain size has something to do with the degree of polyploidy.
Yeah.
Because you mentioned that, to my point of view, is the kind of mixing up between, on the one hand, the degree of polyploidy, and you showed the probability that you will find more polyploidy in C4 grasses, may lead, as we all know, that polyploidy leads to bigger plants and bigger morphologic seeds and so on. And again, I would agree that due to polyploidy, the pollen grain size will also increase. So if this is triggered by polyploidy in the first place, then I have the question, what has polyploidy to do with the C4 syndrome, which is a physiological syndrome?
Okay.
If this is underlying the pollen grain size.
There are two. The first point is my results are specific to my data set. Like, I'm looking at the results only in the 160 species data set I have. My claim is not about the whole Poaceae family growing in all the world. Probably from other regions, contradictory results may come. But And the other thing regarding polyploidy, I have not established the ploidy level of my species. This is already established, and once I identified my species, then I looked at the published data on the ploidy level. And then I divided, okay, this is my whole 160 species data set, and this is the published information on the ploidy level of this species. And on that basis, I made groups, diploid and polyploids, and looked at the effect, and we saw that there were trends that polyploidy has a role, because in both cases, C3 polyploids had greater grain size than C3 diploid species. And in C4 and C4 species also, C4 polyploids had greater than grain size than C4 diploid species, showing that there is an inherent effect of ploidy. What is that effect? That is to be seen. That is yet to be seen. But at least I have established that there is an inherent effect whereby polyploid species has a larger grain size than the diploid species growing in the same climate. So here, if climate has a role, that is you can say in a sense nullified, because in the same climate the polyploid species are showing greater grain size than the diploid species collected from the same grassland growing in the same climate. And moreover, if we compare the differences of the C3 species with the C4, not now ignoring the size of ploidy, but of the photosynthetic pathway, then I again made data sets of my whole 160 species, and I divided them into, okay, C3 species versus C4 species, and what is the result? And there I saw that C4 grains showed trends of larger grain size than C3 species, which indicates that photosynthetic pathway has a role. And here my results stops. I don't have further, like, what is that inherent mechanism that I haven't, that question I haven't addressed. But at least I have established that there is a trend that C4 and C3 species growing in the same grassland, in the same climate, are showing different trends.
So I will remain with the same topic. And so your basic finding was that grain size, falling grain size of the C4 grasses tends to be larger than that of the C3 grasses. My first question is, how new is this information? And the second question is, how useful is it for vegetation reconstruction? Because grasslands can be dominated by a few species. And if you just want to decide if a grassland formerly was C4 grass dominated or C3 grass dominated, and your grain sizes of the C3 and C4 grasses are only tendencies, then it could happen that you find dominant untypical grass, and so you have mixed conclusions. So how useful will this be for vegetation construction, and how new is the information?
Okay, so regarding your first question, how useful it is, like the first thing is, like, the area which I have worked on is the first time, like this is first contribution to scientific community that grasses have been reported from a far-flung area. The second thing is that I have worked with recent Poaceae species, and then we say, okay, if these are our results, can we apply them on past grasslands? And paleoecologists, when they take cores and they study their samples, so Poaceae grains are distinct from rest of the pollen grains. So we can take Poaceae grains from the cores and then look at their increasing or decreasing trend parallel to a certain age. And during that age, then we can see what are the increasing or decreasing trend. And the results we get, we can then compare with the result from phytolith assemblages and isotopic studies. And then we can see in the later stage how much our results match with results from isotopic studies, because different people work and there are people who are interested in phytolith assemblages. We can compare our results with them, and this, I believe that this can then help us in hypothesizing further questions regarding Earth's climate.
Do you think that you will get additional information? It's actually a thing like what Khush asked. Additional information in addition to the isotopic analysis. Wouldn't the isotopic analysis not be enough to divide between C3 and C4 classes? Maybe it would be more exact?
Well, frankly, I don't have much idea about what phytoliths people work or like isotopic people. As a palynologist, my work will be to apply the results of my own pollen studies. But what results I get from my own study of pollen, I can then at least have, okay, what are the claims from those people? And look into my own perspective. From the perspective of a paleoecologist or palynologist, I will look at, like, if my results are contradictory to what they claim or no, or they are the same. And that, I think, is the point where we can hypothesize further questions.
Yeah, well, if you have to stay with the polyploid topic, the first question is technical one. So you said you did not identify the ploidal level like counting. So how did you select the taxon? How did you deal with cases where more than one cytotype is reported in one species? And let's see, how can you be sure that you really have a diploid versus polyploid data set?
Okay, so this was an issue for me, and like as I told that once the species were properly identified with the help of taxonomists using reproductive characters and online search, we came to a conclusion, okay, because grass systematics is quite difficult, identifying grass species, your taxonomists know better. But once the species is there, then we look at the published data on the ploidy level. The diploid species were simple, but the case of polyploid, like triploid, tetraploid, pentaploid, that was then creating problem for me. So I considered all the triploid, tetraploid, pentaploid, and so on, and I made a group and marked them as polyploid. Whether that was the published information about that specific species was triploid or hexaploid or whatever, I considered the rest all of them as polyploid. So I made two groups. I didn't make much groups, two groups, diploid and polyploid, the rest all. And then looked into it.
You should be aware that, of course, for literature research and for it's not really, let's say, final evidence that your sample that you looked at home is actually a diploid of the polyploid problem. So, okay. But what I want to ask further, which relates a bit to the previous questions, when you want to relate this now to your study area, I mean, there are some trends related to polyploid. For instance, the polyploid is more frequent in colder climates, and the diploids are more in the tropical regions and so on. This is quite well known, and there's a lot of literature. But if you are interested in such questions, then why focusing just on grasses? Then it would have been better to make samples across, let's say, bigger families of angiosperms And look here at the diploid polyploid pattern, because the trend is not only in Poaceae, it's in all angiosperm families, in all grains. Polyploids are bigger than the diploids. So then you might have, let's say, some information of, let's say, climatic situation related to polyploid in your area with a simple method.
Yeah, so my selection of this Poaceae family was not only just my emotional attachment for grasses, that I love grasses, but it was also the fact that grasses are abundant, they're everywhere. You don't have to climb 300 meters to them.